we're going to dump out the Windex. We're going to sterilize them real quick in ethanol, let them dry, and then we're going to coat them with something called Fibronectin. So we're going to cover the slides. It's going to make basically a bunch of attachment points for our cells. And then we're going to let those set up for Thursday and for our dishes. So I'm just going to, these were in Windex. All I'm going to do is just dump them. And then I'm going to rinse them a little bit. Normally we would soak this in ethanol a little bit more. But in the interest of time, I'm going to pour a little on these. This is 70% ethanol. Does anyone know what the ethanol is doing? Sterilizing, right. So, you guys saw Genevieve do sterile technique. Obviously, we don't have room to put you all in the hood, right? So we're going to practice doing sterile technique just on the bench. So you're going to do this over your bench, but you're going to spray 70% ethanol in your hands, and then wipe your hands, or like just let it dry, basically. So you're all going to take the spray bottle, spray your gloves, so your gloves are sterile. And then I'm going to go around and give you all 2 slides. So Genevieve, I pulled out the fibronectin. We have, so there should be a spray bottle on every bench, and then 2 people should be at each bench. Okay. So then while I'm getting these slides out, we're going to make our fibronectin. So do you want to explain how to make the fibronectin, Genevieve?
Yeah, absolutely. And I'm going to do it up at the board. So you guys don't have to do this calculation yourself, but we'll review it again next week. Fibronectin, which is one of the proteins that cells can adhere to. So as I mentioned before, they won't stick to the glass, so we have to put something on the glass that they can stick to, and that's fibronectin. Right now, if this is a stock solution, and the concentration is 1 milligram per mil, there's 20 microliters. I'll do this later, and I'll get back to you guys. So we need to put that 20 microliters into 2 mils of PBS. So 20 microliters to 2 mils. That's a 1 to 100 dilution, right?
Yeah, is it not in
20, so this is also 2,000 microliters, right? So 20 and 2,000 is a 1 to 100 dilution. So we're actually making, so the stock solution is 1 milligram per mil, and we are making a 10 microgram per mil solution, I think. I'll check my math later, but I'm pretty sure that's right.
So
anyways, you need to put 2,000 microliters into one of these tubes. And in this case, it's okay to not adjust for this volume. Technically, you would do 1,980 and subtract this 20, but you guys don't have to worry about that. I will caution you though, this tube does not fit 2,000 microliters. It only fits 1,700 microliters. So I would do 1,000 and transfer it to another tube and then dilute it again with 1,000.
do you want to just demo that really fast?
Yeah. So this is a 1,000 microliter pipetter. I set it to 1,000 microliters. I'm going to grab one of these tips. Do we have PBS out?
Yes.
Or did you forget that?
Right.
Fabulous. Okay, so 1,000 microliters of PBS. And I'm going to put it inside this tube. You can see like a pretty tiny volume. I don't know if you can see that. So that 20 microliters is in here. I'm going to pipette this in. And kind of like what you just saw me do with the cell suspension, I'm going to mix it slowly, but a little bit forcefully so that I can stir it well. And then I'm going to pick that volume back up and transfer it to one of these red tubes because these actually will hold 2 mils. So I'm going to take all of this. And I need another, so now we need one more pipette full of 1,000 microliters. And same thing. Just mix, but you want to be careful because this tube is pretty full, so if you mix too forcefully, it'll splash out, and then your volumes won't be accurate anymore. Okay? Can everybody see what I'm doing? So it's kind of like dilute and transfer and dilute one more time. Okay. So what we're going to do with this fibronectin solution is actually coat some of your slides so that we can put the cells on them next week and they'll have something to stick to. So I just want to get all the ethanol off because the ethanol itself is toxic to the cells, right? And we're going to set the slide inside one of these Petri dishes. Literally just set it down on the bottom like that. And then I'm going to take the fibronectin solution, actually take 1 ml, so you can do this for your 2 slides. So again, this was already well mixed. So I'm going to pipette this and I'm almost going to paint the slide with the solution so that there's a nice portion in the center that has the liquid on it. It's okay to like scratch the bottom a little bit, but I'm literally just painting kind of like a tiny rectangle, not tiny, but like maybe the middle 1/3 of the slide. You have a little extra you can touch the edges. So after you do this, you want to be careful not to hit it so that the liquid spills off. There's surface tension on the slide that'll keep the liquid from spilling out of the dish. And this is just going to create a nice layer in the middle of the slide that has that protein that your cells can attach to. So now, not only do we have a place for our cells to grow or to live, but we also have a clear slide that we can do microscopy through. And we can keep the cells alive and image them while they're alive. So I'm also going to, the nice thing about these Petri dishes is they have a lid. So I'm literally just going to set that over here very carefully and cover it. And it takes about 30 minutes for that solution to like stick, and then we'll just suck it up and let it dry and it's good to go. And we'll wrap the dish in parafilm so that it doesn't, the protein doesn't degrade over time, and keep it in the refrigerator so that it's stable, and it'll be ready for us to use either Thursday or Saturday.
You know what I mean? Just like some up and down. Just just to stir it up a little bit. Since it's such a small volume, just doing that is enough to like give it a proper mix. You can go ahead, you can try. Yeah, I mean try mixing it, just give it a little practice, but yeah, then you can do your trick.
Yeah. Yeah, and the other thing you guys can do here, I want to show you. Your reaction when you did it. Since these are
He was so boring. He was just silent, like I ain't do it.
That's perfect. Yeah. You don't even have to take all of it. So there are some cases, again, it depends on the experiment that you're doing. So in this case, we're not really concerned about the total cell number, and we're not concerned about the cell density. So we don't really care.
Here, guys.
Oh, yeah.
Mike,
Yep.
do we have to dry these off?
No, don't dry them off because you want the fibronectin to just kind of settle into it.
She said don't touch it.
You can, your petri dishes, you can label initials and the date because you'll use your slides next time.
Do I make one even though I'm
Two slides, yeah.
Is that it?
Yep, so that can go in the orange tube. And now, get a fresh tip. Yep, get anything else you can out of there.
Everyone has 2 petri dishes? What?
With the solutions that you have to buy. You did those
So you want to create like a nice, like a nice round surface for it to sit on. So you can probably use about 7 of these together. You don't have to be specific though. So go ahead and grab a little bit. And did you want to eat it?
like how much should I put?
Probably like 7 Let's try that. It's just like paint kind of a circle. Oh, I see what's happening. You just got under a little bit. Not a problem. Yeah, that's good. So then here, if you actually touch the tip to the bottom, you can sort of spread it around. No, yeah, exactly. There you go.
Let's find time to talk about step one, but you.
Has everybody coded over here? No. Do you guys have coded yet? Okay. Has everybody coded over here? Someone's still waiting for pipettes. Everyone's Have you already taken?
Yeah, but yeah.
So if we were doing an experiment and we were careful about like the cell density and the cell number, we would have to be very specific about the area that we were giving them to grow.
I don't know.
And the experiment that we're going to do, we don't actually care about that.
What happened to the two I gave you?
Or I guess a different way of saying it is it's not important for our experiment. So actually it doesn't matter if you have like some edges that are exposed, as long as you have a good sense of where the coating is, so that when we go to put the cells on them on Saturday, that you know where to put them. Does that make sense? Yeah. So you're covered because you have a lot.
Did you add in? So you guys have to add in?
It's 1,000.
Okay, so you haven't, you never pipetted any PDS? Okay, where did that?
But I'll wait till these guys are done. So the reason we've made these slides again is because we're going to do an experiment with the cells on next Saturday where we stain them with that calcium indicator dye and we get them to light up like the video I showed this morning. But really to observe that on the microscope we need a clear glass slide to transmit the light through. So we can't do it in the plastic dish or the cell culture dishes that we've been working with because that plastic will fragment the light so much that there's not a strong fluorescent signal. So we have to do the experiments on glass. And this way the cells will be really beautiful and hopefully we'll get a strong signal. So what I'm gonna do to, I was saying to a couple of you guys as I was walking around, for this particular experiment, we're not concerned about how many cells we're going to have on the slide or what density there are at because we're not doing anything quantitative. So it's not like we're trying to compare protein expression between different groups and we have to normalize to anything. We don't need any of that extra information. So in this particular instance, it's not important to control the area that the cells are going to attach to. So it's okay if some of you had like the middle 1/3 of your slide covered, or if some of your solution went towards the edge, as long as you kind of have a good sense of where that sweet spot is for the cells to stick to. And if you don't know, you can always leave a little note to yourself. So like left side was well covered, or even draw a little circle around the area that was kind of like nicely populated. So then in a couple days when we come back to this slide, you have a good reference point for where to put your cells. But again, that part's not important to us. If it were, then we would have to be much more rigorous in the area that we're pulling and the amount that we're using, and we would have to carry that through to when we're using the cells as well. So this part is a lot easier if you have one of those aspirators that I was using in the cell culture hood that has a nice suction and just carries the waste into a container. But because we don't have that, it's not a problem. We can actually use a pipette to do it. And I'm just going to slowly drop the liquid and dispel it into the waste container. And after I get some of it, it's helpful to actually tilt the slide, but you don't want all of the fibronectin to run off the edge and then potentially get underneath the slide. We don't want cells to stick to the bottom of the slide, only to the top. So I'm tilting it kind of gradually. And if there's a little bit left over, it's not a big deal. What you can do, and what I usually do when I do this and I'm working underneath the cell culture hood, is I will let the rest of it evaporate off the top of the slide by leaving, so I don't know if you can see, but there's like just a couple spots left that would be really hard to suck up with this. So you could just tilt the lid so it's mostly covered, but if this were underneath the cell culture hood, that would be like sterile enough and you could let the rest evaporate, and in about 5 minutes it would be clean and then good to go. So I'm just gonna show you the last part. Fibronectin is a protein, so it will degrade, especially if it's not kept in the right storage conditions. So the way that we store these slides is by wrapping them in parafilm so that they can't keep evaporating, and then we stick them in the refrigerator to keep the protein stable. So I'm just gonna take a tiny bit of parafilm. Scissors, thank you. And I actually, because I'm wrapping the edge of this dish, I like to cut kind of a long strip. Get it started and hold it along the edge and just slowly pull it around. Stretch and stretch. Press it to seal. So it's all wrapped up and good to go in the refrigerator. Okay? Any questions? All right. Have a wonderful Saturday evening.